So thank you everyone for coming today. Our speaker today is of course Dr. Eng Kuan Moo. For those of you who don't know him, I'll do a brief introduction. So in 2009, he received his degree in biomedical engineering from the University of Malaya in Malaysia. As has been a theme for a lot of the postdoctoral scholars we've had this year, he jumped right in to do his doctoral studies right after graduating with his undergrad degree. He did it at the same university where he studied biomedical engineering and biomechanics, also at the University of Malaya. Specifically, his project was focused on in situ and in vitro chondrocyte biomechanics. And he did that working with Dr. Belinda Murphy and Dr. Noor Abu Osman at that university, where he graduated in the year 2014. So that's all the stuff I didn't know about Eng Kuan's background. What I did know is that he's been here at the HPL for a very long time.
How long?
Since 2010, so 9 or 10 years, depending on how you want to count it.
But he's in the intern's office.
Yes, and he's been in the intern's office. So he knows that place inside and out at this point, right?
That's true. Someone needs to make sure that everything is in order.
Yeah. So basically, Eng Kuan, he started as a summer student or international intern, and then after he graduated, he's been a postdoctoral fellow here with us for all this time. What that means is that it's really, really difficult to summarize all of Eng Kuan's research projects and research interests, because he's got a whole lot of them, but I'll do my best. He's mentioned two particular fields he's very interested in. The first is tissue mechanics, so where he looks at the behavior of articular cartilage in response to loads, mechanisms of cartilage degeneration, as well as the distribution of sarcomere spacing in whole muscles. So both cartilage and muscle work he's interested in. He's also done a lot of stuff involving cell mechanics, so looking at cell morphology and how that changes in response to load, cell membrane properties he's quantified, and he's also studied, he's done studies looking at the mechanisms of death. With all of that in mind, when I asked him kind of what his most proud research accomplishments are, he said he's really been proud of the fact that he's been able to kind of move in and do all of this work involving muscle, but still kind of keep up with his original studies, his original field, which was in cartilage mechanics. With that said, I think he should be just as proud of the fact, if not more, that he recently at ISB this year won the Promising Scientist Award. And I think many of you guys, I saw you there, who saw his talk. So I think that's a really great accomplishment too, by the way.
Thank you.
Of course, it's not all about work. So outside of work, Eng Kuan, like many of the people we've had here, likes to do a lot of outdoor activities. And of course, being in Calgary, being near the mountains, gives him lots of opportunities. He's mentioned that he really likes mountain biking and hiking, which are amazing summer activities, but he gets a little bit depressed during the winter time, which really makes me think that as Canadians, we failed him, because we clearly haven't introduced him to skiing or snowboarding. So maybe we should get on that. He's also said that he's recently interested in bouldering, and he enjoys solving bouldering problems. I don't know what that means exactly, but if you're as good at solving bouldering problems as you are at solving research problems, I think you'll be great at it. So with that said, I'll pass it on to you.
All right. Thank you, Ifas. So, good afternoon, everybody. Thank you very much for spending some time with me today. I'm going to talk about in situ imaging of sarcomeres in skeletal muscle by using, imaging technique to derive the biomechanics. So skeletal muscle are the biological tissues that power our daily movement through voluntary contraction. They consist of a few structural levels, going from the whole muscle level to fascicle, then to the muscle cells, and then to the subcellular structure called myofibril. Depending on how you look at them, skeletal muscle can have a very different appearance, as exemplified by the cross-section and the longitudinal sections shown here. From this longitudinal section, we can tell that the skeletal muscle is striated, and these striation patterns contain important structural and functional informations of the skeletal muscle. We'll focus on this small region highlighted in yellow in the next slide. Under bright field microscopy, the muscle contains repeating patterns of light and dark bands. If we zoom in this yellow box, we can visualize the most basic contractile unit of the muscle, that is sarcomere. A sarcomere is defined by the region between two Z lines. It contains light isotropic band, or I band, that are made of actin filaments, and dark anisotropy band, or A band, which are composed primarily of myosin molecules, but also allow actin filament to slide in. According to the cross-bridge theory, the actin filament and the myosin heads need to interact with each other for force production. Now, if we zoom in further in this orange region, this is exactly what we expect to see: the interactions between the actin filament and the myosin heads. More than half a century ago, Gordon, Huxley, and Julian came up with the sliding filament theory that can predict muscle force based on the structure of sarcomeres. They proved this theory through a series of isometric contractions of isolated muscle fibers. Now, this is the classic sarcomere force-length relationship of a skeletal muscle, derived from the sliding filament theory, with the y-axis being the muscle force and the x-axis being the sarcomere length. It describes the maximal force capabilities of a muscle, which uniquely depend on the amount of overlap between the actin and the myosin filaments. The force length curve has an inverted U shape and consists of the ascending limb, plateau, and the descending limb, and can be derived theoretically from the lengths of the contractile proteins, actin and myosin filaments. Typically, myosin length is constant at 1.6 micrometer in vertebrates, while actin filament length is species dependent and varies between 0.9 to 1.2 micrometer. But within a species, the myosin and actin filament lengths are normally constant. Now here's a quick tutorial on how to construct theoretical force length curve using the lengths of the contractile proteins. Now let the myosin and the actin filament lengths be the blue and the green lines with 1.6 micrometer and 1.1 micrometer respectively. There are a few transition points that mark important events of the sliding movement between the actin and the myosin filaments. Imagine the myosin to be centered and static, and the actin filaments slide inward to the center. We will start from the right of the descending limb, as indicated by the red dot. Now here the actin and myosin filaments just loose overlap, and the force is 0. And the length can be obtained from the summations of 2 times actin filament length plus the myosin filament length plus the Z-line thickness. Now as the actin filaments slide inwards over the myosin, the overlapping area increases and therefore increases the force. And the force will reach the maximum in this configuration. The reason that the force length curve has a plateau is because of its structure. Let's look back at the electron micrograph of a sarcomere. Notice in the middle of the sarcomere, there's no available myosin head to interact with actin. Now, it is called the bare zone, and it is about 0.17 micrometer wide. For that reason, there is no force change when the actin filaments slide over this region. Now, at this point, the sarcomere length is the summations of 2 times actin filament length and the thickness of the Z-line. And when the sarcomere gets shorter than this length, the opposing actin filaments start to interfere with the actin and myosin interactions of the other half of the sarcomeres. As a result, the force starts to drop. When the sarcomere shortens to the length of the myosin, when the sarcomere lengths get shorter, when the sarcomere is shortened to the length of a myosin, which is 1.6 micrometer plus the Z-line thickness, the Z-line begins to compress the myosin filament, causing the force to drop at an even faster rate. Eventually, the sarcomere will produce zero force when it reaches about 1.27 micrometer. Now, with all the values of the transition points determined, we can then construct the theoretical force length curve. The Sarcomere force length relationship was established using isolated muscle fiber, shown in this yellow region. It has also been shown to work at the myofibrillar level. But the question is, would it work at the whole muscle level? For decades, the muscle functional behavior has been taught as upscale versions of a single sarcomere. This is an attractive idea, considering that the sarcomere is about 3 to 4 orders of magnitude smaller than the whole muscle, and therefore the total numbers of sarcomeres within the whole muscle is enormous. For example, in this 30 centimeter long tibialis anterior, there are more than 100,000 sarcomeres lining along this TA length. Most of our knowledge of scaling from single sarcomeres to the whole muscle is gained from cadaveric studies, which assume that the mean sarcomere length measured at a single spot and at a given muscle length under relaxed conditions represents the sarcomere length across the entire muscle. However, this simplifying assumption has not been tested systematically before. In fact, there were a few attempts in the past to reproduce the sarcomere force length relationship at the whole muscle level using cat soleus muscle. And we will do a quick review of the results from these studies. Now, here's the theoretical curve, force length curve, built based on the myosin and the actin filament length of a cat. The yellow region represents the theoretical plateau region, and the Reck and Westbury in 1969 measured the plateau region to be at a longer length, which lies between 2.8 to 3 micrometer. And this means that the force length curve should be to the right of that theoretical one. On the other hand, in 2012, Vaz et al. Found that the plateau region to be on the left of the theoretical curve and was twice as wide as the theoretical plateau. In short, these studies suggest that the experimental force length curve measured experimentally did not follow the theoretical one at the whole muscle level. It's not surprising that the sarcomere force length curve may not apply at the whole muscle level. Looking at the structure of the whole muscle, there are a lot of confounding factors that could deviate the sarcomeres from behaving according to the theoretical force length relationship, which was established initially using isolated muscle fiber. To name a few of these factors, the muscle fibers in the whole muscle are surrounded by extracellular matrix, arranged in a complex muscle architecture and mechanically connected in series with elastic components and therefore the associated compliance. And also, the sarcomere length distributions in the whole muscle is not uniform. And depending on the theoretical model used, the relative contributions of passive and active forces in the total measured force can be different. In the next few slides, I will talk more about these factors. First, the muscle fibers are mechanically connected in a parallel arrangement with the adjacent muscle fibers by the extracellular matrix. This image shows the connective tissue within the muscle after all the muscle fibers were digested away. It is quite clear that the thickness of the connective tissue is not evenly distributed. So this suggests that the contraction behavior of one muscle fiber can be influenced by the adjacent muscle fibers to a varying degree, depending on the surrounding extracellular matrix. Second, the muscle has a complex architecture. Let's take this human tibialis anterior as an example. researchers have used MRI diffusion tensor imaging to investigate the arrangement of muscle fascicles in the muscle, represented by these pink lines. Now, if we zoom in this yellow box, and you can see that most fascicles do not run the whole muscle length. And also, they run at an angle to the longitudinal axis of the muscle, which we call the pennation angle. In addition, the muscle thickness varies when going from the proximal end to the distal end, thereby changing the amount of force produced at each cross-sectional plane. And third, in an intact muscle tendon unit, the muscle in red is connected to a series elastic component, in this case the free tendon at the distal end, in yellow. When the muscle contracted, the compliance of the free tendon can lead to muscle shortening, even when the muscle tendon unit is held asymmetrically. The relative length change of the muscle fibers and the whole muscle is shown in this graph, with the y-axis being the fiber length change and the x-axis being the muscle length change. You can see that the muscle length change was always larger than the corresponding fiber length change. Not only that, the relative change in length between the muscle, the whole muscle, and the muscle fiber is also dependent on the activation level. The yellow curve shows the passive stretching behavior, and the red curve shows the corresponding length change when the muscle was maximally activated. The difference between these two activation states can reach up to 10% of the optimal fiber length. A few years ago, we found that the sarcomere lengths in a small region of relaxed muscles were very dispersed. For example, in the mid-region of the tibialis anterior, the sarcomere length can differ by up to 0.7 micrometer. Functionally, such a dispersed length suggests that the corresponding force can differ by up to 20%. It is currently unknown of how this sarcomere length non-uniformity affects the functional behavior of the skeletal muscles. And finally, the representations of muscle architecture in theoretical modeling can influence on how we interpret how the muscle works. For example, there are two popular heel-type models present in the literature. On the left shows the standard model, and on the right shows the structural model. In the standard model, the length of the parallel elastic component depends on the muscle length, while in the structural model, the parallel elastic component depends on the sarcomere length. So therefore, during muscle contraction, the force productions from the parallel elastic component will not change in the standard model, but will decrease in the structural model. With so many confounding factors and experimental verifications of the sarcomere force length relationship at the whole muscle level is urgently needed. But due to technical difficulties, sarcomere contraction behaviors have never been studied in situ in maximally contracting muscle tendon units. Therefore, the goals of the current study was to address this question and to determine experimentally the active sarcomere force length relationship in an intact tibialis anterior of mice, and to compare the experimental force length curve with the theoretical one. Also, we aim to compare the standard and the structural heel model to determine which model is suitable for describing the contraction dynamics in the intact whole muscle. We hypothesized that the average sarcomere lengths measured from a tiny volume of muscle mid-belly can predict the muscle force according to the theoretical force length curve. Also, we hypothesized that the structural heel-type model is a better model than the standard one for the modeling of a muscle tendon unit. Before we start, let's take a look at the anatomy of the tibialis anterior of mice. Here's a drawing of a mouse hind limb, with the left being the knee and the right being the foot of the mouse. the TA is highlighted in yellow. If we fillet the muscle longitudinally, the TA will look something like this. A schematic diagram of the sagittal view of TA shows the fan-like muscle fibers orientation and their attachment points on the tendon plates. Since TA is opaque, we can't see through the muscle and investigate the striation patterns. In order to visualize the sarcomeres, we use state-of-the-art multifocal microscope, shown in the green box here, in order to perform second harmonic generations imaging of the TA muscle. High-intensity near-infrared laser was used as the excitation source. The interaction between the laser light and the TA drew light signal from the myosin molecules within the sarcomeres. The emitted light, the light signal, was then collected through the same objective lens by a photomultiplier tube at a harmonic frequency. Here's a representative image of a muscle. The bright band represents the A bands. To process the muscle image, I crop a region of interest containing 15 to 20 sarcomeres that are arranged in series. The gray values of the image was plotted along the longitudinal axis, as shown in this graph. The image intensities changes sinusoidally along the longitudinal axis. After filtering the image, the centroids of each sinusoidal curve was determined and marked by the green asterisks. Sarcomere length was measured from the distance between adjacent centroids. For the experiment, we performed a simultaneous recording of sarcomere images and muscle forces during activation. Muscle forces induced by supramaximal nerve stimulation were measured using a miniaturized dynamometer, while the corresponding sarcomere images were taken using second harmonic generation imaging. Here's a representative example. On the top is a force trace over 1 second, and the bottom shows the corresponding sarcomere images at different time points. We first take a snapshot of the sarcomeres in the relaxed muscle at time 0. Then a 600 millisecond activation period was applied, and the sarcomere images were taken at time points indicated by the green boxes on the time axis. During the experiment, we measured the total force exerted by the TA muscle, which include the passive part and the active part of the muscle force, as highlighted by the green curve here. As the sarcomere force length relationship describes the active muscle force, we need to determine the passive force and then subtract it from the total force in order to get the active force. The blue curve shows the passive force length curve of mouse TA. When the muscle is stretched to a target length, the passive force increases. At the target length, the muscle was maximally activated and the in situ sarcomeres shortened. For our analysis, we used the standard and the structural Hill type models to determine the passive force. According to the standard model, the maximum passive force prior to activation is used, and we call it apparent passive force. The structural models takes into account of the shortening of the sarcomeres during the contractions, and we call this corrected passive force. After data analysis, here are what we found. We first look at the sarcomere lengths and the muscle forces in bar graphs. On the left is the mean sarcomere length, and on the right shows the muscle forces as a function of muscle length changes. The muscle was stretched from its initial length of 10 to 11 millimeter by 0.5 millimeter to 4 millimeter. In the relaxed muscle, this will lead to an increase of mean sarcomere length, as well as in an increase in the muscle force. In the activated muscles, the mean sarcomere length also increases in length, but the muscle force initially stays constant, but then decreases with increasing muscle stretch, which resembles the descending limb of the force length curve. One thing to point out, when the active force was higher than the passive force, the activations of the muscles led to 5 to 8% shortening of the sarcomeres. However, when the opposite occurred, meaning that the passive force was higher than the active force, there was no activation-induced shortening of the sarcomeres. Next, we plot the sarcomere force length curve using the experimental data collected from 6 animals and compare the results with the theoretical one. 3 graphs are placed side by side. On the left, we plot the apparent active forces as a function of sarcomere length measured in the relaxed muscles to see if sarcomere length measured from cadaveric studies can be directly applied to infer functional behaviors of muscles. In the middle, we used the standard Hill type model and derived the apparent active force as a function of active sarcomere length. And on the right, the structural Hill type model was used to derive the corrected active force, and they were plotted against the sarcomere length measured from the activated muscles. Next, the theoretical force length curve is plotted for reference. We found 2 actin lengths in the literatures that differ by 25%. Since we're not sure which actin length is the correct one, we plot them both to see how they match with the experimental data. Now the data of the first animal was overlaid onto the graphs, and each data point represents the mean sarcomere length and force. The horizontal and the vertical lines shows the standard deviations of sarcomere lengths and muscle forces, respectively. Next, the data points from the remaining 5 animals are included. It is obvious that the experimental data better fitted the theoretical curve constructed from actin filament length of 0.95 micrometer. It turns out that the 1.2 micrometer actin filament length was derived from neonatal animals, and the actin filament shortens after the first week of birth. 0.95 micrometer actin length was derived from the age-matched animals. So we get rid of the blue theoretical curve. To evaluate the experimental data, we best fitted a linear line of negative slope to the data on the descending limb, as illustrated by the dotted lines here. It is clear that the data on the right graph fitted the theoretical curve the best. And we show that the sarcomere lengths measured from the activated muscles and the active muscle force determined using the structural Hill type model best match the theoretical curves. To summarize, first, we found that the sarcomere lengths measured in the relaxed muscles cannot predict the force capacity of the muscle, as the resulting force length curve is shifted to the right of the theoretical curve. Second, the structural Hill type model is better suited than the standard one in describing the mechanical behavior of intact muscle tendon units. And third, actin filaments shorten throughout the lifespans of the animals and should therefore be obtained from the age-matched animals. Finally, sarcomere lengths measured from the mid-belly of the activated muscles can predict the relative muscle force according to the theoretical force length curve. So, is this the finished stories? Not at all. Our results only represents the sarcomeres in the mid-belly of the muscle. We picked this region because previous cadaveric studies have sampled sarcomeres in this region. The fact that our experimental data fitted the theoretical curve so well may have to do with the local architecture at the mid-belly of the TA. Likely, the line of actions of the muscle fibers in the mid-TA coincides with the line of actions of the whole muscle, which reproduce the experimental condition needed that was used during the isolated muscle fibers experiment. However, the sarcomere behavior in the distal end, proximal end, and even the deeper part of the muscles are still unknown to us. I think that with our study, it should stimulate other researchers to further investigate the in situ sarcomeres by studying each of the 5 confounding factors that I mentioned earlier and see how sarcomere contraction dynamics are affected by these factors. One important question that arose from the current study is that what is the effect of sarcomere length dispersion? We found that the sarcomeres in series can differ in length by up to 0.6 micrometer. In our daily movement, the muscle goes through a range of motion. Now, when the muscle shortens, the sarcomere dispersions may shift entirely to the ascending limb, and the resulting force can differ by 30%. On the other hand, when the muscle length increases, the sarcomere dispersions may shift entirely to the descending limb, leading the force difference to be around 40%. Although this is a hypothetical scenario, it points to an important fact that we need to gain more understanding on how the sarcomere length non-uniformity affect the functional behavior of whole muscle. In conclusion, the sarcomere force length curve can be applied at the whole muscle level. We show that the average sarcomere lengths measured from the individual sarcomere lengths within the muscle midbelly can predict the muscle force capacity of whole muscle according to the sliding filament theory. In order to construct the theoretical force length curve, the lengths of the contractile proteins, especially the actin filament lengths, should be obtained from the age-matched animals. And finally, there's still a big question mark over the role of sarcomere length non-uniformity in muscle functional properties. Now, with that, I would like to thank everybody who is involved in this study, especially my supervisor, Dr. Walter Herzog, and I'm open to any questions. Thank you very much.
Awesome. Thank you very much. So I'll open up the floor to any questions.
Nice talk, Eng Kuan.
Thank you.
Do you think that the amount of non-uniformity in sarcomere length is consistent across muscles?
No, not that from our measurements. So 3 years ago, we measured the sarcomere length non-uniformity across the entire muscle of tibialis anterior of mice, and we found that the sarcomere length in the proximal end is more dispersed compared to the one in the medial, lateral, and the distal end. Yeah.
But then, so say would a TA exhibit the same properties as like a soleus muscle? Like, would you see the same kind of patterning?
I'm not sure about that. I don't think people measure that yet. But it's an interesting question to see if the sarcomere length non-uniformity actually depends on the muscle fiber type, right? I don't know about that.
Or like the function of the muscle. Does a muscle that goes through a greater range of motion exhibit more non-uniformity than a muscle that's active in a very small range of motion?
That is, yeah, that's fair. I don't know about that. I think we should dive in more into this area and just investigate different muscles.
Right.
How did Marco Voss compare, or does anybody compare, the whole muscle work that's been done previously, and then like put the force length relationship in terms of like what's going on at the sarcomere level?
So what people have been doing was that they tested on the whole muscle, the force versus the muscle length changes.
And it's just animal models?
Animal models,
Yeah.
Cat's And then what they did is after that, they fix the animal, the particular configurations, and then they tease out all the muscle fibers and use a laser diffraction technique to measure the sarcomere length.
Okay, so they actually get some measures of bits of sarcomere length.
Yes,
that
yes.
relate it back to the
Yeah.
muscle. I see.
So most of the time, people use isolated muscle fibers by fixing the joint at a particular configurations, and then later on they say, okay, this sarcomere length corresponds to that particular angle of the joint, so they associate it with the functions.
I see.
Mhm.
If I could ask another question. One place where I was a little bit lost is where you're talking about how you calculate active force contribution.
Mhm.
And you have the total force subtracting the passive force.
Yes.
And then you talked about these different hill type models that helped you do that. But in my mind, I'm thinking, why don't you just measure the force when you stretch it passively and subtract it from the force when it's activated?
Yeah,
Why do you need the hill model?
I can show you that. This one. So in this one, the passive force contributions is mainly accounted for by the parallel elastic component, right? So for the standard hill type model, the length of the parallel elastic component is given by the muscle length. So which means that whatever you stretch prior to your activation, that will be a passive force. But if you look at the structural hill model, it is only in parallel with the contractile components, which is the sarcomeres. And the series elastic component is not in parallel arrangement with the parallel elastic component. So which means that during the activations of the muscle, of an intact muscle tendon unit, the compliance of the free tendon at the distal end will lead to a shortening of the whole muscle. And therefore, the length of the sarcomeres will change. And you have to take into account of that by using the structural hill model. That is what the standard hill model cannot do. Right?
No, I think I get it. So it's really just so you can get the lengths associated with a particular force.
Yes,
Yeah, okay.
that's
Thanks.
correct.
Kind of silly definition question. When you say muscle length, do you mean muscle plus tendon, or do you mean between the two ends of the muscle without the tendon, not including pronation, or
I mean the fixed muscle tendon unit length. Yeah. Yeah.
The data points that you showed, those bars, were those the variability?
Which
So
one?
If I saw it correctly, it looks like the variability increases as the sarcomere length increases.
You mean this curve?
Yeah, it seems like the variability as sarcomere length kind of increases tends to increase.
You mean this one?
Yeah, all of those on there compared to the ones before that are shorter, right?
Yes.
So why would that be?
So I calculate this curve using individual sarcomere length. So if you can see, the passive force length curve is very steep. So when I'm on the descending limb, which means that I'm on this area, a slight change of the sarcomere length can lead to a very big differences in the passive force. And that's why it leads to that huge standard error bar.
You were talking about how the actin length changes with age, at least in between neonatal and the age you have. Right?
Yes.
Do we know if that continues throughout life, or is that just an early development then? And can you summarize for me what that means for you as like for your muscle production ability? Is that a good thing or is that a bad thing as you age?
Yeah, that's a very good question. So initially when we started this project, like what I show you here, we got a value from an electron microscope study, which is the 1.2 micrometer that we got. So it was like far towards the right of the theoretical force length curve. So when we put our experimental data, it was completely off. So then we deep dive into the literatures and see what's going on. Like it doesn't make sense because, like I said earlier in the presentation, the actin filament length and the myosin filament length within a species should be relatively constant, right? Then we found out, oh, actually the actin filament length can shorten after the first 2 weeks. And then I think this study is done by Gokin et al. From California. They found out this behavior, and then they also tracked the actin filament length for up to past the adult stage into old age, but then they didn't see a huge changes of the thin filament length. So the changes mostly was found after the neonatal age. So functionally, I don't think it will changes the function that much. I understand that by changing the thin filament length, then you shift the force length relationship level rightwards. But this is not happening in the real animal. In the slow twitch fibers, actually they found some changes when you're getting older. Yeah, not the fast twitch fibers type that is like TA.
What you did basically depends on actin and myosin.
Yes.
Actin doesn't play a role. Now, you know, the last 700 presentations here are all about Titan. And
Yeah.
now you come and talk about muscle without mentioning that.
So that's a very good question, actually, Beno.
Thank you.
The Titan filament plays an important role for the muscle force productions, especially when the muscle is stretched to a long length. And nowadays, like recently, like 2015, group I think it's Linari et al. Published in Nature, they found that the thick filament can actually senses the different stretch or the stress level that is applied to the sarcomeres. And with that, it changes the amount of cross bridge that is involved in the force production. So Titan is very important, but not when you're on the ascending limb or plateau where you haven't changed or stretched the Titan yet. But at a later stage, like longer length on the descending limb, then its role started to come in and really stabilize.
Most of your points were on the descending.
You're right. They are very important. But with our technique, we didn't look into that effect of Titan. I can see that we can use some drugs to, destabilize the Titan molecules and see how it affects the functional properties. That could be a future work. That can keep me for another 10 years.
What's your understanding of the reason of the phenomenon of sarcomere length non-uniformity?
I don't know. That's my answer to that phenomenon.
That's speculative.
What I think is that the sarcomere length non-uniformity is a nature design. Nature is not perfect, and therefore it will not give you the perfectly uniform striation patterns within the muscle. But then nature has a different. Well, I once listened to this TED talk. They would say that nature is like tinkering with what is available. So throughout the evolutions, they add a different increasing functions of a different design to add for that imperfect design in the beginning. So in this sense, Titan may play a role trying to accommodate that imperfect design. But in general, I have no idea why the sarcomere length uniformity exists. But the interesting thing is that the average behavior of the sarcomeres can be predicted using theory. That is the part that we know.
Would you expect the thin filament density to be the same for every single sarcomere? Or the myosin density, number of heads, I don't know, whatever, however you guys talk about it. Would you expect it to be the exact same for every single sarcomere?
That is likely the issue that causes sarcomere length non-uniformity. No, I don't expect
Hey, I
them.
just solved it.
Yeah. Actually, Walter, if I remember correctly, Walter was trying to do a study using electron microscopy to count the amount of contractile proteins per cross sections. So going to different area of the sarcomeres, or going to a different sarcomeres within one single myofiber, trying to count the number of contractile proteins to see it corresponds to that sarcomere length non-uniformity. But it's a very technically challenging project, so it was not done yet.
But I think you have to assume that that relative density of actin myosin influences the quality of the sarcomere. I don't want to know what you want to call it, which just affects. So with that being said, and knowing that there's just sort of these inherent differences in quality between sarcomeres, like, I'm always surprised when you guys talk about a sarcomere and you say the plateau starts at 2.4 micrometers and it goes for 0.17 micrometers and the descending limb finishes at 3.6 micrometers or whatever.
Yeah.
So is that just theoretical, and on average we should see this type of behavior? Because it's really impressive. The data that you showed is like really, really impressive. It really follows that theoretical behavior for 6 different animals.
Yeah. It's all actually there are some dispersion there, but in general they follow the theoretical curve. But yes, I have the same questions the first time that I get into this field. I listened to Walter talking about, oh yeah, this is at 1.6, like 0.17 micrometer wide bare zone. Really fascinated me. That's why I'm in this field, trying to understand what's going on. And sure enough, we found that the average behavior of the sarcomeres goes just like the theoretical predictions, which is surprising. But if you go to individual sarcomeres, then the whole thing doesn't work anymore. So why is that? Why would we have these kind of imperfections in nature design? It's hard to understand. So it puts to the theory which model the muscle as the upscale versions of single sarcomeres into question. So can we do that and predict like single sarcomere behavior within the whole muscle? Or should we just step back and then start from a different idea and go from there? It's just hard to answer.
This is from Walter.
Hi, Walter.
He has 2 questions. Question number 1 for you is that, do you think your result from mouse TA can be generalized to other muscles?
No, I do not think so. Just like what I answered to Bex, we don't even know if the behavior is the same at the distal end, at the proximal end, or even at the deeper part of the muscle. So what we found is that only in the mid belly of the muscle it works. But how about the other parts of the muscles? So that's why we need to investigate this area more, and using different types of muscle like soleus, or mainly fast twitch fiber type muscle like gastrocnemius, a different pennation angle to see the effect of that.
So question number 2 for you from Walter. So when people do work with single fiber and sarcomere length are not controlled, then the plateau is much wider and the force much greater than predicted theoretically. Why did that not occur in your case?
I don't know. Can you repeat the question again? I'm not sure if I understand it
He
correctly.
said that when people do work with single fiber and
Yes.
sarcomere lengths are not controlled,
Uh-huh.
then the plateau is much wider and the force much greater than predicted theoretically. Why did that not occur in your case?
Yeah, that's a very good question. I remember Jerry Pollack presented compilations of all the literature's values, and they found that the plateau is much wider and the values at the descending limb is higher than the theoretical predicted. Why did I not see that in my study? I have no idea. It could be that it happened, I pick a right region of the muscle and then investigated it, and then it happened that it coincides with what the single muscle fiber produce. In the contradiction is that most of the work that was presented before was done using single fiber, which is a better experimental conditions than the whole muscle level. I don't know. I believe that the muscle fiber type has a role to play. Yeah, that's all I can speculate. I have no idea.
And my apologies, he says to ask question 2 first before question 1.
What's question 3?
You say, Benno already mentioned it, so disregard question number 3.
The title. I see. So, is there any other questions? Yes, Rob.
In a whole muscle, we'll start the other way. When you're doing single muscle fiber, you've got the sarcomeres lined up in the direction of the force. When you've got a whole muscle, you've got muscle fibers, they're on curves and they're in different parts of the curves. And so in a way, you've got a variable pronation angle, even on a single muscle fiber. I haven't done it, but if you model that, does that, for a start, blend out the sharpness of the transitions on the length tension curve from the hill model, where you have those sharp turns and onto the plateau? And does it make does it sort of blur the whole thing by no longer being in a straight line? So if you perhaps modelled a single fibre going round a bit of a circle, going round the belly of the muscle, have you tried to model what influence that would have?
No,
Because
actually.
you're comparing it with one that's in a straight line.
Oh, you mean the descending?
Yeah, well, on the whole length tension curve, for a single sarcomere, that's done in a dead straight line on a single fibre. And in the whole muscle, the muscle
It's curved,
fibres
yeah.
traverse around the belly and in all various different architectures. So you kind of expect it to at least blur out those transitions. And for that sake, for someone as clever as yourself in modelling, to just even take a single fibre and make it go around an artificial belly, what does that do?
The short answer is that I don't know. I haven't done modeling in muscle yet. It could be in the future study, but the reason that we see in this behavior could be that we only measure the sarcomere length at a very local area.
Yeah.
So within that local area, the muscle fibers can just run in parallel with the line of actions of the whole muscle. And that's why it reproduces what people have found in the single fiber experiment. So I expect the fibers at the distal end with a curvatures, with a pronation angle, and the fibers in the proximal end that that has a different orientations could behave differently.
But even if they had the same uniform sarcomere length all the way along the muscle fibre, their representation in the force in the XY direction, in the X direction, could be variable. These ones are contributing, that are perpendicular, that are in parallel with your line of measurement, versus the ones that are perhaps going around the belly. So they've only got a cosine component coming in the direction that you're measuring.
Yeah.
So you might interpret what's actually a different angle, you might model and interpret it as, well, it must be a different Sarcomere length in terms of its contribution.
Yep, it could be. I know that Dr. Blanker from Virginia University, her famous work was in modeling the fascicle strands within the whole muscle. And what she found that because of the pennation angle and the orientations of that, different lengths, because the fascicles doesn't run the whole length of the whole muscle, that's why it leads to a non-uniform strand distributions within the fascicles. So yes, the architecture plays a huge role with the whole muscle functions, how the sarcomeres or fibers interact with each other. Yeah. But personally, I haven't done anything with modeling of skeletal muscle. Maybe I should.
I'm just following up on Rob's question.
Yes.
You showed early on a picture where they were able to measure, I forget if it was, or they were able to image fibrils or fibers, I forget.
Oh, which one?
Really early on, you showed a nice picture with lots of lines and lots of different directions.
The
Basically,
MRI.
Yeah.
Yeah.
But basically, my question related to that is
Yeah.
sort of piggybacking on what Rob's saying. Has anyone ever tried modeling the muscle as a structure, as a combination of sarcomeres that are attached to each other in a certain way? And does that influence the result that you get? Does it maybe a model that's better?
Could be, yeah. The more the information you have with the structures that you put it into the model, I will believe that it can improve the model. Yeah. Any other question?
Yeah, it seems like we are. Lindsay, did you have? No. Okay. No, looks like we are out of questions.
Thank you very much.
So next week we'll be hearing from our own Dr. Brett Edwards, who's going to be talking on the importance of early therapeutic treatment to prevent bone loss after spinal cord injury. So I'll see you all there.